Good morning, everyone. I am Moa Kalaf, a postdoctoral research fellow at the University of Ottawa Heart Institute. I hope everyone is healthy and safe at this time, wherever you are. Thank you for joining us on this virtual science seminar series brought to you by the BEaTS Research and Radio team, continuing to bring science to the community. Some housekeeping notes before we get started. The seminar will be 30 to 40 minutes of uninterrupted presentation, followed by, there we go, followed by a 10-minute question period. Today, I will be your seminar moderator. I'm Mo. Please keep your microphones and camera off during the presentation. If you have issues connecting to the audio, you can call in by phone. Please refer to the email sent to you after registration for phone numbers and meeting ID. For all questions, please send in a private type chat my way. I will ask our guest speaker all questions after the presentation. Include your name, title, institute, or program affiliation whenever possible. Seminars will be made available on the BEaTS Research Radio YouTube channel soon afterwards. Check our website and Twitter for release information. Today, I have the pleasure of moderating an exciting lecture by our guest scientist, Dr. Milica Radisic. And next week, we have our own Dr. Emilio Alarcon giving a seminar. So stay tuned for that. So just a brief introduction about our speaker today. Dr. Radisic is a professor at the University of Toronto and Canada Research Chair in Functional Cardiovascular Tissue Engineering. Dr. Radisic obtained her PhD from MIT, Massachusetts Institute of Technology. She's received numerous awards and fellowships over the years. The long-term objective of Dr. Radisic's research is to enable cardiovascular regeneration through tissue engineering and development of new biomaterials. Her research interests include microfluidic cell separation and development of in vitro models for drug testing. She is also a co-founder of a startup company called Tara Biosystems, focused on the use of engineered tissues in drug development. Thank you, Dr. Radisic, for joining us today. Welcome, and please take the virtual podium for the seminar.
Thank you very much for the opportunity. So the field of organ-on-chip engineering is a pretty new field. It just started in basically year 2010 when Don Ingber published his landmark paper on lung on a chip. And if you look at the numbers of papers published each year after that, it is clear that the field is on the exponential increase, whereas the field of microfluidics is still rising, but not as fast as organ-on-chip engineering. And so many devices have been published and described, but if we try to categorize them, we usually see emergence of three types of devices. One of them are these interface-on-a-chip devices that basically mimic epithelial-endothelial barrier. And lung-on-a-chip device, like shown here, belongs to that group. It will basically be a membrane with epithelial cells on one side and endothelial cells on the other side. And there are many other approaches, such as those for vascularization, etc. Impressively, very, very large number of companies that are trying to commercialize these interface-on-a-chip devices, including Emulate, which is one of the leaders in the field. Another category are really devices that are focused on mimicking liver or heart on a chip. And the reason, and we call it just the things that are focused on mimicking this parenchymal tissue on a chip. And the selection of these two tissue types is really driven by drug toxicity. So these two tissue types are always very affected. They are number one and number two cause of withdrawal of already approved drugs. And if you look here, there are a number of cardiac systems. Ours, for example, is called BioWire, and there is tissue patch also that could be used for drug testing. And there are many systems that include two posts that the tissue is attached on, as well as these cantilevers pioneered by Kit Parker's lab. A large number of these companies is also on the in the process of commercializing these technologies, including Tata, that has already commercialized BioWire tissues from our lab. One area that is probably the most difficult to break into is this area of body on a chip. And that is the idea that we can take different compartments, let's say heart, bone, kidney, liver, gut, and connect them through joint circulation. And there are a number of approaches described, including some of our own work, where we're just connecting two organs in series. The difficulties of this approach include the fact that we don't know how to scale, and very often there are different culture media requirements, and it's difficult for us to predict like how small the heart should be in relation to area, because let's say the lung, because in the body the size of the organs is perfectly matched according to metabolic activity, blood residence times, masses, and surface area. And when we try to make devices like this, we usually pick one criteria, let's say metabolic activity or blood residence time or surface area, but then the others don't fall into place perfectly. But this is also an opportunity to develop scaling rules through these approaches. And commensurate to the amount of the effort, the number of devices that are being commercialized through this approach is less. But the overarching hypothesis behind all the organ on a chip work, I would say, is that tissue size and tissue structure and vascular structure are all important, so that better function will follow better form. And towards this goal, I'm going to talk to you about some of our technologies, most of which incorporate vascularization, which is really essential for organ-on-a-chip approaches. And several years ago, Boyoung Zhang, who is now a professor at McMaster University, he came up with this concept. He said, why don't we go to the clean room and use the techniques in this microfabrication industry to create branching networks of polymer on a chip. And basically, he calls this angio chip. And so by 3D stamping technique that he developed, he would create branching vasculature, as you can see here, out of polymer. And then it's actually just branching polymer vessels. And at the end, these get endothelialized, and that's how a vasculature is created. So you can see branching in X, Y, and Z direction. Polymers are usually not permeable to larger molecules, to proteins and cells, but we render them permeable by incorporation of these nanopores and microholes. And also, by that enables extravasation of the cells from the vasculature to the parenchyma, or movement in the opposite direction, intravasation, such as in metastasis. And what we can see here is that basically these spaces in the parenchyma are interconnected, so they enable cell sealing, and they can also be patterned in such a way to enable matching of the mechanical properties to that of native tissue. And so although this approach is manual, it's still scalable. You can create many angio chips in one shot. And if you look here at the footprint of a 96-well plate, we are able to fit 3 tissues in it, 3 angio chips. So we drive flow by pressure head. So this was one of our requirements. We wanted to eliminate the use of pumps to make it easy for people to use our systems. And so when you add in the upstream well compared to the downstream well, you will get a pressure head that will drive flow. The first step is definitely endothelialization, followed by seeding in the parenchyma, and then gel compaction that results in the formation of the tissue. And you can see that this endothelialization can really be nice. In this particular case, these are CD31 positive endothelial cells fully covering the lumen, are migrating through those microholes that I mentioned at the beginning. And this is really the first step of angiogenesis. As they start migrating, then permeability to small molecules will really go up, because in this system, it is the endothelium that controls permeability. We were also able to demonstrate that we can do study phenomena such as monocyte rolling on the vasculature, and especially if we activate the endothelium with TNF alpha, we can see a lot more monocytes rolling and extravasating through microhole where basically they are now. This approach, we can vascularize heart tissue, and we call this heart and plate because of the well plate setup. And we can use both rat and human cells. We can see that the blood vessels are present, the tissue is twitching, it's propagating electrical impulses, and we can see that we can affect the beating rate of these tissues by adding drugs such as epinephrine or digoxin. Well, similarly, using human embryonic stem cell derived hepatocytes or rather, this liver will is positive for albumin, and it's also secreting urea. Interestingly, it processes drugs. If we add a drug such as terfenadine to the inlet well and we let it perfuse through the vasculature, we can collect that drug in the outlet well. But in this case, we're collecting hexafenadine, which is the process of results from the metabolic processing. So we can see that the terfenadine was delivered to this liver on a chip in a very physiological way, where we had both convection and diffusion to be able to reach the tissue, as well as reaction or turning over in the liver space into the product of metabolism, hexafenadine. This platform also enables us to scale up to create really thick tissues. They're about 2 millimeters thick as the left ventricle of the rat, not human yet. So this is about 2 millimeters. And to achieve that thickness, we also had to centrifuge the cells through the scaffold uniformly, and we seeded CD31 positive endothelial cells in the tissue space as well as in these lumens to demonstrate the interaction and better cell survival through the thickness. The angiochip is unique in the sense that it enables direct connection to the host vasculature, and you can see that here by basically inserting inlet into the artery, and this is artery bypass configuration. We can establish immediate flow, or we can see artery to vein configuration here, where we see basically an AV shunt loop being created. Angiochip is still pretty big for disease modeling and drug testing because it uses almost 2 million cells. Despite its relatively small size, the cell density is actually very, very high. So we wanted to scale down, and we created this platform called INVADE, Integrated Vasculature for Assessing Dynamic Events. And so in the INVADE platform, we have a blood vessel, which is made out of polymer, the same polymer we use for angiochip, which is called POMAC, and it's spanning the entire length of a well of a 96 well plate. And so then basically you need 3 wells to run one tissue. You need one inlet, the middle compartment for the tissue, and one outlet. And by doing this, we also have the opportunity to connect these multi-organs easily in series. So here you can see how we were able to model liver, heart, and tumor, all using the common vasculature. What's interesting about angiochip, and you can see here, angiotube, we call this tube angiotube. It also has these microholes that are very important for cell extravasation and intravasation phenomena. And you can see this one tube, which becomes a blood vessel, lining being positioned in the middle of a microwell. And what you can see here in the SEM image is a smaller well within a 96 well plate, right? So that's what it looks like. And not only that, but in the heart chamber, we also add these cantilevers on each end. So the movement of the cantilevers will enable us to track force of contraction. In addition, we can incorporate electrodes, carbon electrodes, into these 96 well plates to enable electrical pacing. So you can see an example of a vascularized cardiac tissue that is beating, the cantilevers are moving, and that's how we are able to track the contraction, both the force and frequency, by looking at the movement of the cantilevers. In this particular case, the drug epinephrine is added through the central lumen that is endothelial. So we really have physiological delivery of the drug. INVADE, the platform, also allows us to create a human liver tissue. As you see here, the tissue is vascularized. And similarly to the previous case, it produces urea and it metabolizes drugs that we're putting in. Using the INVADE platform, we were also able to study elements of cancer metastasis. And so what you can see here is just a breast cancer model. The cells are GP positive, and we can basically detect shedding of these metastatic cells into the outlet well, and we can see how these cells are increasing in number with time in culture. But not only that, but we can actually visualize, not just count the number of cells, but actually visualize this entire process by capturing metastatic endothelialized blood vessel. So this is from the breast cancer tissue. We can see many metastatic cells in the lumen. And in this particular case, we connected this tissue to the heart tissue. And in the heart tissue, you will be able to see a few metastatic cells lodging into that compartment as well. So these metastatic cells were able to transfer epithelial barrier twice and lodge in the downstream tissue. But interestingly, also a good chunk of the tissue is devoid of these cells. So this is a very interesting question for us to study why this particular cardiac tissue is able to basically prevent profound metastatic invasion. But in terms of this idea that, we have organoids on a chip on one side versus organoids on the other side. The organoids are basically developed by biologists, and they enable us to create miniature, like spherical structures by directed differentiation, usually of adult or human pluripotent stem cells. And so they have really nice cell fidelity, but they don't have such good control over flows in and out, right? And so we published a position paper a few years ago speaking about this idea that, with organoids, you have really beautiful cell fidelity, but you really don't have any flow. You don't have ability to instrument these organoids, nor the ability to mechanically or electrically stimulate them. And really, organoid chip wins there. But organoid chip is still relatively low in cell fidelity. There is a very common use still of cell lines and not enough different cell types that make a tissue. So just to show that we can, move in a new direction and really marry organoids with organoid chip engineering, we started working with patient-specific pancreatic tumor organoids. So this paper just came out in Advanced Functional Materials in a recent issue. So what we did here is we took our INVADE platform that are described in the previous slides, and around an endothelialized vessel, we would seed single cells derived from primary pancreatic tumor from patients who were operated on in Toronto General Hospital. And so we can use their specific pancreatic organoid media in the parenchymal space here, because one of the features of our platform is that the endothelium and endothelial media and parenchymal media don't mix. So what happens there is, with time, you can see that these initially single cells really grow and they turn into individual pancreatic tumor organoids. So you can see that the tissue is actually surviving very well in this platform. But a more interesting question is what happens when you add fibroblasts? Because in the native environment of these tumors, pancreatic tumor is deadly and really doesn't have good prognosis at all, with the survival of under 1 year and from diagnosis. And the contribution of these cells in the stroma, such as fibroblastic cell types in the body, contributes to development of very dense matrix that basically shields off the tumor cells from therapy. And so what we did here is we compare the properties of this tumor on a chip model that's constructed with human tumor cells in the form of organoids, with and without supporting stromal cells. And so the control was obviously just fibroblast tissue that didn't have any tumor. And even if you just glance at this, you can see that in this co-culture where tumor is co-cultured with fibroblasts, these individual organoids are larger and they're much more densely packed. These organoids do have open lumens, and they're positive for Ki-19, CK19, which is a marker of epithelial cells. But also, if you look in this co-cultured tissue in between organoids, you can see a lot of alpha smooth muscle positive, alpha smooth muscle actin positive fibroblasts. So the tumor itself speaks to the fibroblasts to turn them into myofibroblast phenotype, and you can see how they are now really secreting a lot of collagen, resulting in the stiffening of this matrix. Now, if you look at this collagen area from second harmonic generation images, there isn't that much collagen in fibroblast alone or in organoid tissue alone. And if you look at the amount here, which is really obvious in the images, there is a lot more than simple addition. If you just add the amount here and the amount here, you're now going to get this graph, which speaks to synergistic effect of this co-culture, where really the presence of tumor cells is making these fibroblasts secrete a lot more collagen than if they were by themselves. And this definitely turns into higher tissue stiffness. This is measured by AFM in situ. And the result of this increased stiffening and increased presence of collagen is now that small molecules cannot permeate into this tissue space effectively. So if we now perfuse small molecule like CFDA, shown here in green, through the endothelialized blood vessel in the organoid tissue, just organoid without fibroblasts, both of these cases are vascularized, you can see that in 4 hours, basically the cells have picked up the dye. However, in co-culture, barely anything has passed in 4 hours, speaking to the hindrance of this transport process. But not only that, this process actually then results in very detrimental effects on our ability to deliver cancer drugs. So if we use a standard chemotherapeutic agent in this co-culture settings with the perfusion through the vasculature, you can see basically no effect on the tissue. And the drug itself, it has been reported to be more selective for cancer cells, so it won't be killing normal human endothelial cells and fibroblasts as much. And if you compare that to the standard organoid tissue culture, as shown here, right? People are now trying to use organoids for drug testing, and they would use them in a matrigel, in a well plate, for example, or in some other matrix. As you can see here, if you do apply the same drug in a dose response, that drug will kill the tumor cells in the organoids, in the dish. But at that same concentration, it won't kill tumor cells that are growing in a vascularized environment supported by fibroblasts. And so basically, this speaks of the ability of organ-on-chip devices to capture that interaction better, and hopefully in the future allow us to discover more effective cancer drugs. Okay. So, and lastly, I'll just speak a little bit about our kidney on a chip system. So kidney diseases, they really start with podocytes, right? And this progression is usually not detectable until you have lost a lot of capacity. So it's number one on the wait lists, and it's number one in terms of the transplants. And it's third largest reason for drug withdrawals from the market. And basically, these podocytes have very specific geometry that you can see here. They're interdigitated, and these interdigitations enable one to create barrier function, enable basically creation of barrier function and selectivity. So what you can see here, we try to reproduce this environment just by seeding podocytes on angiotube scaffolds in the InVade platform. But we didn't get much. They were basically just pancakes. And then we told ourselves, well, let's look better at this structure, because they're really podocytes are sitting on a curved surface. And this curved topography is really really, enabling them to create proper interdigitations. So we hypothesized that if we just provide simple curve topography to these podocytes, we will get improved phenotype. So we cultivated podocytes in bubble versus flat substrates. And we saw that irrespective of what's in the culture media, on the flat substrate, we always got less nephrin, which is a key molecule in slit diaphragms that compared to the bubbles. So topography itself was definitely, very significantly, by two-way ANOVA, we can determine effects of the factors. Topography by itself is very significantly enhancing the expression of nephrin. And when we then quantify this interdigitation ratio in a different media, we see that on the bubble surface, we really approach the interdigitation ratio that we see in the native kidney. The gray area is native kidney. And again, we see very significant by two-way ANOVA influence of topography. People often tell me that pictures like this are just philopodia. So we took some other cell, like mesenchymal stem cell, and drew them on the same surface and tried to look at branching. And we never see these many levels of branching using other cell types. So it's definitely, these are philopodia. Some of these might also be philopodia, but the levels of multiple levels of branching that we see here would be indicating that we are moving towards interdigitations, although we are not there yet. So what Anastasia Kotter for my lab did is she created these membranes that had topographical features, right? So they had topography, but they were also permeable membranes. And she would place them in a transvaal insert. So this is a very simple configuration where now you have at the bottom podocytes, and at the top you can see the endothelial cells if you want, or you can leave this blank. And you can see that topography by itself enables us to create improved barrier function. So despite having more surface area here, we actually see improved barrier function on the surface with bubble topography. And this now enables us to study the effect of cytotoxic drugs such as PAN. And eventually, we think that we can really move to other areas as well that depend on similar spherical topography. Another question is, well, what do we do next, right? Can we enhance these topographical features through more refined topographical cues, such as something that's more fractal, such as found in the native kidney glomerulus? And so this idea is that we can take a slice of glomerulus and pattern it to improve this topography further. But how do you move from something that's 2D into 3D, right? And the ideal is if we had the perfusible structure that incorporated topography. And because most of the kidney work is done on flat membrane, create perfusible structure. That's something that Rouja, my lab, did. This is published in Advanced Materials in 2018, but the H fiber technique she just published in ACS Central Science. And so not only that she needed to have this alginate fiber that is perfusible, this alginate fiber had a knot. And on top of the knot, there were additional microtopographical cues. So we have topography on top of the topography to mimic as much as possible the spherical cues coming from the kidney glomerulus. So to generate this perfusible knotted fiber, so she would basically use alginate deposited in a bath with calcium, but so she would also incorporate calcium carbonate and expose to acid, so that when these CO2 gases escape the bubble during the cross-linking of alginate, they create a topographical structure, such as shown here. So you can see this knotted fiber with topographical structure, and it's all on top of a soft material such as alginate. So Rouja was able to place this entire setup in a 96-well plate platform that is very similar to our Invade platform. The actual footprint is from the Invade platform. And you can see the fiber is perfused, it's knotted, and we only have selective flow really through the main lumen. It's hard to see on a very small structure such as the fiber, so she used hanging drop technique to create very uniform cell density on the entire fiber. And through these measurements, she was able to demonstrate that both the knot and topography are really important, because if you just look at the flat tube, some people may question, why do you need this when you have a tube? If you look at the flat tube, you can see here very, very clearly, here in SEM images, that these cells are all to align, and that's not the topography you want to have in podocytes. This is something you will have in muscle cells, but not at all in podocytes. And whereas if you look here on plus knot and plus topography, we have really beautiful branching of the cells and interdigitation as well. It is also possible to endothelialize this inner lumen, and we show here that we have normalized permeability to bovine serum albumin that is really good by podocytes, as it should be. We also showed that smaller barrier for weight, the range where in terms of permeability, which is a harder number than just normalized transfer, as we can see here, we can see here very clearly that bare H fiber has pretty high permeability, but cellularized H fiber has almost no permeability to albumin, and it's actually within a range of what you would expect for permeability for normal glomerulus. And so I'd like to say that our work at the University of Toronto is really enabled by a group of fantastic people. We have this facility that we call CRAFT, which is a collaboration Center for Research and Application in Fluidic Technologies. It's a collaboration with NRC, National Research Council, and it really contains state-of-the-art equipment for microfabrication that allows us to do this. Obviously, we are still shut down because of COVID, and the access to equipment will probably be slower, but it would not be possible to do any of this without this equipment. We also have excellent trainees that we are training through Answer Create training program in entrepreneurship and organ-on-chip technologies. Basically, our trainees are already spinning off companies. They started several like TissueX, Cohesis, Cutera, etc. And it's really the goal to enable kids from and we are awarded equipment through very generous CFI grant. And so in summary, I showed you several of our technologies, perfusible angiochip and Invade platforms. They don't use any pumps, no bulky boxes, and no connectors. They're compatible with current pharma equipment, and we saw perfusible heart and liver tissue for drug testing, cancer model for study of metastasis. I also spoke a bit about top membrane region and H fiber, which allow us to establish barrier function on podocytes. And so at the end, I'd like to thank everybody in my lab who helped with this work, our funding sources, and I'll be happy to take your questions now.
Thank you so much, Dr. Radisic, for an amazing conversation about the future of where this is all going. I have a couple of questions here, if you don't mind. So yeah, the camera's on. So the question here is, what are the main challenges with body-on-chip systems, and what approaches do you suggest to address these issues?
So as far as I understand it, scaling is the biggest challenge, right? So if you're the value of these systems is so that you can study how the drugs distribute through the body, right? PK and PKD. But the problem is, what are you going to choose as a criterion to put them together, Are you going to look at the mass? Like there is in your body, like you don't have a brain that's this big and like this small liver, right? That it's all perfectly scaled. And also this scaling could also be personalized or individualized, right? It's not always identical in individual to individual. And so what do you choose as the criterion? Because if you choose surface area, then you'll be off maybe for mass. And the underlying reason for this is that we still cannot vascularize tissues perfectly. If we were able to vascularize them perfectly and achieve that perfect cell density, all of these parameters would fall into place ideally, but that's still not possible. Another limitation is really, do you have relevant maturity and availability of relevant cells for all of these different compartments? So in some fields, we are more further along. There was a lot of work on cardiac maturation, but some others might be a little behind, like podocytes. There's only a couple of protocols published to even differentiate them from iPS cells, right? So those are some of the limitations in trying to create this whole body of genes.
So thank you so much. The next question is, when you think about solid tumors, those are normally encapsulated within a fibrous layer. This case is in part responsible for reduced efficacy of chemotherapeutic drugs. Could the organ on a chip becomes a strategy to mimic such an environment?
Yeah, that's exactly why we were doing this work, right? And basically, and we used the pancreatic organoids from the patients, because if you use cancer cell lines, it's basically very different game. And we only put 10% fibroblasts there, versus if you look in the histologies of pancreatic tumor, by volume, most of it looks like fibroblasts. There's like little specks of pancreatic tumor spheroids in a sea of fibroblastic matrix. And even just with, it looks like there is 90% by volume in histologies. When we do our experiment only with 10% of fibroblasts, we basically completely abolish the effectiveness of this chemotherapy. I think this is something really important for us to include in the screening experiments, and I would not say that every single candidate drug, a handful of those that are most promising candidates, I strongly argue to be tested in these kinds of this dense matrix may not be sufficient. It's actually, this is a bigger deal, much bigger deal for immunotherapies, because everybody is so excited about T cells being able to kill cancer, but this shield of collagen basically means that they can get to cancer and kill it. That's actually probably even more exciting question to statistician to answer this. I apologize. I don't know why my connection is unstable. I got the best possible internet that one can get the fiber. And it's fiber optic and it's still unstable.
That's all right. This is the world we live in now. So the next question I have is so specifically maybe addressing the idea of how do we prioritize and what best fits. The question is, the nodding approach you showed for kidney is quite fascinating. Could you use a similar approach for producing lung-like structures?
Yes, of course. I mean, it is possible. The drawback is that alginate fiber and that knot is still pretty thick, right? We're still basically in the epithelial and endothelial barrier. There is 10 microns between two cell types, right? There is the distance is so tiny, and here we have about 100 microns. So it's larger distance than in the body. And so that is the limitation of that system. However, you can't make that much smaller because then the gel itself, you won't be too fragile, you won't be able to lift it up. But yeah, that's what we are hoping, that these topographical systems, basically systems that include bubble surface or bubbles on top of knotted fiber, that we can use them in other areas, including lung or lymphatic tissues, everything that depends on that topography.
All right. I think with that, we'd like to thank you for coming in today and fighting through the internet to give us a wonderful seminar. Thank you so much, and thank you all for attending this seminar meet. And we'll see you next week. Thank you.
Okay, great. Thank you very much. Thanks. Bye-bye. Bye.